I'm just going to walk you through. Oh, I've got the focus wrong. Oh no, I think it'll be alright. I'm just going to walk you through a couple of questions that are, some of them, the first one's reasonably straightforward, the second one slightly less so, and then we're going to have a go at something that's a little bit more unfamiliar. The key thing with genetics questions is to read the question properly. I know it sounds really obvious, really straightforward, and you'll be astonished at how many students clearly just try and do a cross that they've done before, the one that they like best. I don't know what it is. So, guinea pigs we're looking at. In guinea pigs, the allele for black coat, black, we're told to use the letter big B, is dominant. And to the allele for albino, which is little b. And, so that's a bit of a dead giveaway. We're now going to be dealing with dyhibrid, because we've got another characteristic. Allele for rough coat, big R, is dominant to the allele for smooth coat, little r. So far so good. So we've got two characteristics, coat colour, coat smoothness, and we've got alleles B and R. A heterozygous black, so heterozygous black, that tells us that it's big B, little b. Smooth coated guinea pig, so smooth is recessive, that must be little r, is mated with a heterozygous rough coated, heterozygous, two like different alleles, coat, rough coat is dominant, big R, little r, albino guinea pig. So that immediately tells us that we're dealing with little b. Complete the genetic diagram. Okay, that's nice and easy. To explain the expected ratios of the genotypes and phenotypes. So we want genotypes and phenotypes, but it's going to tell us what to put where. So parental phenotypes. So these are the ones that we've mated together. So we've got a black, smooth guinea pig, crossed with a, and I'm going to put the colours in the same order now as well. I'm going to put the characteristics. So I'm going to say that this one's an albino rough guinea pig. Just to make my life easier, and it will then follow down to the genotypes. So parental genotypes now, so these are my letters, I've now got to combine them up. So black we were told was heterozygous, big B, little b, smooth, little r, put my cross sign in so I don't get confused. Albino, little b, rough coated, heterozygote, big R, little r. I then need to do the genotypes of my gametes. So I need to foil now. First letter, big B, little r, put a circle around it, it's one gamete type. Outside, big B, little r, got one, don't need to write it down again. Little b, little r, different sort. Little b, last letter, little r, that's the same. Put my cross sign in again, so I'm keeping my parents separate. First letter, little b, big R. Outside, little b, little r. Inside, little b, big R, got one of those. Last, little b, little r, and I've got one of those. Now that simplifies my Punnett square, so I don't have to draw 16 boxes. Parent 1, big B, big R gametes, little b, little r gametes, little b, big R from parent 2, little b, little r. Combining them then, my first offspring is big B, big R, little r, little b, big R, little r. So remembering to keep my B's together and my R's together, but my capitals first, just makes it so much easier when you're deriving your phenotypes. So, genotypes of offspring. Offspring 1. big B, big R, little r, and we want to match that to a phenotype. So that is going to be big B black and big R rough. Then we've got one of those. Well, I'm hoping we've only got one. Little b, big R, big bar. So little b albino, little b, big R, little r, albino, and big R rough. Then we've got big B, little r. This is going to be black. I'm running out of room. Smooth. And then we've got little b, little r, albino. I'm just going to squeeze that in underneath. Smooth. So I've matched up my genotypes with my phenotypes exactly, and you must make sure you do that, otherwise it makes a nonsense of the whole thing. So that will give us a 1 to 1, they're all different phenotypes, to 1 to 1 ratio. 4 marks. Excellent.